Hello, everyone. My name is Nabin. I'm a postdoctoral scholar at the Computational Bioscience Group here at the Scientific Data Division. My topic for today is multimodal AI models for computer microscopy and correlate the pre-study towards biology molecules. Uh, to give you a context, we are living through a revolution in structural biology. AI-based methods like AlphaFold predicts the three-dimensional atomic structure of proteins, um, which made to the highlight of the paper in Nature. Uh, Image on the top right shows the mechanism of our works. The input is a one-dimensional amino acid sequence, and it predicts a three-dimensional atomic structure from the sequence with high accuracy. Uh, Below are some of the examples of the structure predicted by AlphaFold, where the protein structure is the experimentally determined structure, and the blue structure are the computationally generated structure. So overall, AlphaFold helps us to predict the static structure of the protein with very high accuracy. However, the proteins are not static in nature. They are dynamic molecules that change their shape as they are performing different functions in our bodies. For example, one of the protein called Guide protein, shown in the image left here, adopts different conformational state during different times of the day. When we run AlphaFold, default AlphaFold predicts only fold state. However, we want to understand how this protein works and how it performs different functions. We need to understand different state of this protein. One of the experimental method that help us to identify this dynamic state of the protein is called cryo-EM microscopy. It helps us to understand how this protein changes shape, or how this protein opens and closes when it binds to different molecules, small molecules, ligand molecules, or different drug targets. However, the data set of cryo-EM is problem is very ill-posed inverse problem. First, talking about the forward model on the waveform stuff, we have given a probability distribution of different conformational state of a protein. If we have atomic structure, it becomes easier to simulate density value for each atom present into the atomic structure and generate a density map, fill it with noises, and finally get a two-dimensional images. This problem is comparatively singular today, comparatively easier than the inverse problem where we are working backwards. Given the two-dimensional particle images, our problem is to reconstruct a three-dimensional density map, which is known as the reconstruction problem in cryo-EM microscopy. Second state is to build the atomic structure that fits locally into this density map. In today's talk, I'll be talking about the problem of building atomic structures from density map. First problem is, given this density map, we want to build the atomic structure that is shown here in a ribbon diagram. So density map data structure is in 3D volumes at voxel grids. The output we are looking for in the 3D coordinate system. Over 40% of the map that's available in electron microscopy data bank does not contain associated atomic structures because the problem is difficult as the density map contains lots of noises, experimental artifacts. There are some missing density values in local region of the density map. And the atomic coordinates that we're trying to build here typically contains only 10% of the density map's volume. Density map could contain millions of voxels, but the 3D coordinates can hundreds to thousands of those atoms. To fill in the gap, we developed a method called Cryo-EM. Cryo-EM is an AI-driven method where the input is a three-dimensional atomic structure, three-dimensional density map, and the RNA sequence. The output we're predicting or generating is a 3D atomic model that fits locally into the density map. The research question that we're trying to answer from this problem is how much structural information can we extract from density map alone without adding any information from protein language model, from templates, or predicting structures. The image on the right shows the overall architecture of Cryo-EM, where given the density map, we have two blocks, deep learning block and the alignment block. First, deep learning block contains common encoder that extract the features of density map and CNN decoder that It takes the atom type and the residue data. That information is fed into alignment model that contains customized data marking model and a third algorithm that then finally generates the stable variable structure. So how well this method works? We compare this method with most widely used method out there from Phoenix team across 128 different density maps. Across all the plots, blue dot represents method developed by us, Cryo-EM. Red dot represents the model built by Phoenix. The solid lines are the regression lines. The shaded region are the 95% confidence interval. In plot A, we're trying to show how well Cryo-EM performs better than Phoenix in F1 score, which is a harmonic mean precision and recall. The second plot B shows the protein-specific metric, which is a template modeling score. Higher the score, the better. Cryo-EM outperforms Phoenix on that. On image C, the plot C, we have a quality score that takes into account the completeness of the structure as well as the precise matching of the atoms, where Cryo-EM performs better. Cryo-EM not only provides the coordinates, it also provides the confidence score per residue. You can see how well the confidence score matches with the known structure. The image here shows a protein structure where yellow is the known ground truth structure, and the model predicted by Cryo-EM is shown in color spectrums going from red to blue. Darker the blue color, it's a high confidence area. The darker the gray, low confidence area. If we were to look at the solved figure A, we can see the structure predicted by Cryo-EM fits very well with the known protein structure, and that is faithfully counted by the confidence score. However, if you look at the solved figure B, we can see there is some deviation happening between the ground truth structure and the model structure built by Cryo-EM, which is again captured properly by the confidence score, where we can see some red reason, local reasons. But in the image D, we can see some of the reasons that are not accurately modeled, and the confidence score picks that up. This is typically happens because the experimental density map might have missing some density values over there, and it could also be some experimental artifacts that is making difficult for the modeling task. For that, we need to have some multi-node model to fix that problem. The overall for this work is we developed an AI-based folding automated method that generates atomic structure from the density map. We validated across different metric on widely used method called Phoenix. Cryo-EM performs better than Phoenix. Future direction we have that we're currently working on is conformational landscape of the protein and the multimodal model that incorporates different data modalities. The project that is ongoing at the Berkeley Lab is LBRT from this project where we have different data modalities, sequence structure, experimental data could be Cryo-EM stacks, functional repressions. And some of the tasks that are enabled by this multimodal model is protein folding, prediction of structure given the sequence, inverse folding, prediction of the sequence given the structure of the protein, a model building that we just discussed, building the 3D atomic structure from the experimental data. Also multimodal modeling where we want to predict the structure from different data modalities, such as sequence, experimental data, function annotation. How do we do all these tasks in one single model? The image on the right shows the overall architecture of our model where we have different data modalities. And we train this model in mass range strategy where we mask out or delete some of the tokens and we allow the deep learning method to get the tokens back. So this helps the model to understand the grammar of the protein, help us to predict, reason, and generate different biological outcomes.